All right, continuing on. The last narration covered section 4.10 on diffusion and also on membrane structure. I also talked about this slide, facilitated diffusion, but I'm a little out of sync here. My reading questions and my worksheet are for this section begin with facilitated diffusion. What do I do? Okay, well, let's go over it again quickly. So just so I don't skip over the slide. Maybe I should ask you questions. You tell me about facilitated diffusion right now. Go ahead. What is it? How does it work? So, what does it mean to facilitate? It means to help. Okay. And why do some substances need to be helped across membranes? Because of their because of their charge. They can't get across. And so certain transport proteins are there to help or facilitate the diffusion of these substances from one side of the membrane to the other because they cannot get through the phospholipid bilayer on their own. So there's two kinds of transport proteins that are involved in facilitated diffusion. One type are channel proteins that have a corridor or tunnel through them. That's like aquaporins that allow water to cross through membranes. They're channel proteins, so water naturally moves from where it's more highly concentrated to where it's less concentrated, going through these channels in the channel protein. But then there's also carrier proteins. And how do they work? What do they carry? They're carrying molecules that cannot cross the phospholipid bilayer. And so on the side of the membrane where the molecules are more highly concentrated, they're bumping into these carrier proteins and sticking to them. And then the carrier proteins move the substance across the membrane and let it go. And then the protein goes back to the way it was. And then another molecule bumps into that carrier protein, it moves it across, so it's moving the substance from high to low concentration, carrying it across. Then there's this disease called cystinuria, where a person does not have the correct transport protein or carrier protein to move the amino acid cysteine and some other amino acids across the membranes of kidney cells, and this leads to kidney stones. So this disease, like many other genetic diseases, has to do with an organism's inability to make a specific transport protein to help move substances across membranes that need to be moved. And in some cases, if that substance can't be moved, the organism can't survive. So there's the quick rundown of facilitated diffusion. The main thing we're going over in this part of the chapter is section 4.12 on osmosis. So what is osmosis? That's pretty straightforward. It's the diffusion of water across a selectively permeable membrane. That's all it is. It's just a term, a word given to the diffusion of water. Because water in all cells of all organisms on the whole planet needs to be able to move and diffuse across cell membranes, and biologists use this term osmosis for convenience to describe that process of water's diffusion. But let's look at this a little more carefully. Water molecules surround dissolved solutes. They would be dissolved solutes because they're polar and water is surrounding these polar molecules like sugars, or ions, water surrounding ions, okay? And by water molecules surrounding these charged substances, that reduces the number of free water molecules that are moving around on their own. And so the side of the membrane that has a lower solute concentration will have a higher concentration of free water molecules. And so water molecules are going to move from the side of the membrane where there's more free ones moving around to the side of the membrane where there are fewer free ones. And that movement, this is down the green text on the slide here, that movement of water molecules from where they're more highly concentrated, freely moving around, to the area where they're in a lower concentration, not as many free ones moving around, that is osmosis. So the figure on the slide here is showing you an apparatus, like this U-shaped test tube kind of thing. And in the middle of that U-shaped apparatus, there's a membrane, a selectively permeable membrane that allows water molecules, free water molecules, to move across, but it doesn't allow the solutes to get across, and water molecules that are surrounding those solutes, they're not going to cross. So what it's showing you is that if you have a higher concentration of solutes on one side of this membrane, so look in the upper left of this figure, there's more purple circles on this side than there are on this other side, then that means there's going to be fewer water molecules that are freely moving around on this right side compared to the left side. So there'll be more free water molecules on the left side where there's less solute. So the water molecules, where there's more free water molecules, they will move across that membrane. So look at the bottom part of the figure where it's kind of magnifying and blowing up the picture. There's more free water molecules on the left side of that membrane than on the right side. So the water will diffuse, osmosis, diffuse across that membrane from left to right, from where the water is higher to where it's lower. And that's all osmosis is. So as long as you understand that, you're good for osmosis, understanding what it is. A more challenging thing is to use the terms that are coming up on the next slide: hypotonic, hypertonic, isotonic, and how water responds to those environments. So let's move on to that next topic. Okay, so this is effect of tonicity on osmosis. So tonicity has to do with the amount of solutes in a solution. So think of like tonic water. I don't know if you're familiar with tonic water, but it has solutes in it, minerals in it to give it flavor. So tonic, solutes. Tonicity has to do with the concentration of solutes. So what effect does the concentration of solutes have on the movement of water going across membranes? That's the focus or topic of this slide. Because the concentration of non-penetrating solutes, those solutes that cannot cross a membrane, determines the direction that water is going to diffuse or move by osmosis. Okay, some definitions. A hypotonic solution is one that has a low concentration of solutes compared to a second solution. Hypertonic solutions have a higher concentration of solutes compared to another solution. And isotonic solutions have equal concentrations of solutes. So hypotonic, hypertonic, isotonic. Let's dissect those terms a little bit more. Again, tonic, think solutes. Hypo. Hypo means less than or below. So if somebody is hypothermic, they fall into a cold, icy lake and their body temperature drops dangerously. Hypothermic, low temperature. If somebody is hypoglycemic, that means their blood sugar is low. How about hyper? Hyper means above, higher. So if you're hyperactive, then you're moving around a lot. If, well, not necessarily moving around a lot, but anyway, hyperactive. How about hypertension? Hypertension is when you have high blood pressure. So hypertonic means high solutes, okay? And iso just simply means equal. Let's look at the figures here. What happens to a cell that's placed in a hypotonic solution? Okay, so if the outside solution is hypotonic, that means the inside of the cell in comparison would be hypertonic. So if the outside of the cell is hypotonic, that means it has lower solutes. And if it has lower solutes, then that means in comparison to the other side of the membrane, it has higher water. And the water will move from where it's higher to where it's lower. So it will move from the hypotonic solution to the hypertonic solution. And in this case, this figure is showing red blood cells, like in your body. And if a red blood cell is placed in a hypotonic solution, the water will move into the cell and the cell will blow up like a balloon and explode and experience what's called lysis. It'll split open. What happens if a red blood cell or any animal cell is put in a hypertonic solution? Well, that means the solution it's put in has a high concentration of solutes and therefore a lower concentration of water compared to the cell. That means water will move out of the cell by osmosis and the cell will shrivel. Bad news. If a cell is isotonic compared to its outside environment, then water will move back and forth equally because not only are there equal solutes on each side of the membrane, but there would also have to then be equal amounts of water. And water will move back and forth equally, and the cell will be stable, and it will neither experience lysis nor will it shrivel. So your body works to maintain your blood, water, and solute concentrations so that they are basically isotonic compared to the cells of your body. And which organ in your body helps to regulate your water and solute balance so that everything is kept isotonic? Those would be your kidneys. And by the way, the process, I want you to know this, the process by which organisms control their solute and water balance is called osmoregulation. Osmoregulation. So your kidneys in your body are helping you to osmoregulate, to control your water and solute balance. Let's take a look at the picture on the bottom of the slide. It's of a paramecium, a little single-celled pond creature. And it's just one cell. So it can't have kidneys. Kidneys are organs. So they use a specialized vacuole called a contractile vacuole. It fills up with water because these creatures live in fresh water. So listen to this carefully, the inside of their cells is hypertonic compared to the outside of the cells in the pond water. So therefore water diffuses into their cells from the pond water, and if they didn't have a way to get rid of that water that's diffusing inside of them from osmosis, they would lysis, they would explode. So they fill up these vacuoles, these big bubbles of membrane with water. They're called contractile vacuoles, and they send the vacuole to the plasma membrane where it fuses, and then exocytosis, it releases the substance to the outside, the water to the outside. So that's also osmoregulation. Little single-celled creatures are also osmoregulating. So to summarize this slide, there's a lot of important stuff here for you to know. Solutions. Understand what it means for solutions to be hypotonic, hypertonic, or isotonic. And understand that that translates to water moving into or out of cells depending on whether the cell is in a hypo, iso, or hypertonic solution. So you'll need some practice with some of these terms, and again, I've got some additional questions and assignment for you to work with this. So Okay, moving on. All right, so I did make kind of a little home video with a whiteboard to explain the concept of tonicity, hypo, hyper, isotonic, and how it works, how water moves into or out of cells based on the tonicity of the cell compared to the tonicity of the outside environment. So this video is uploaded to YouTube. You should be able to access it here with this link. Hopefully, fingers crossed, and watch the video and then move on to the next slide. Okay, hopefully it'll help you to understand this concept a little bit better before you move on. Okay, so honestly, what happens to plant cells under different tonicities isn't quite as important to me that you understand as compared to animal cells. And that's mostly because you're taking this biology class, most of you, to go into the health sciences. Humans are animals, so I really want you to understand what happens to our cells as animal cells in different tonicities. But let's just briefly look at this. For plant cells, the situation's a little bit different for what's healthy for them, because plant cells have cell walls around them. So do bacteria, prokaryotic cells. So do fungal cells. They all have cell walls around their plasma membranes, and the cell walls help to prevent the cells from rupturing and experiencing lysis. So plant cells actually do the best in a hypotonic solution. And for us, our cells, if they're put in a hypotonic solution, they're going to explode, lysis. But plant cells, they're in a hypotonic solution, and water goes into them, osmosis, and then the plasma membrane pushes against the cell wall and kind of expands it a little bit, and that's called turgor pressure. And that's how plants are able to stand upright, is turgor pressure inside the plant cells helps the plant to stand upright. If you don't water your plant for a while and the soil gets dry and the plant is struggling to get water, then the cells, the plant cells that are in your plant, are not going to be surrounded by a hypotonic solution because there's very little water available, and it might be isotonic. And that's what the middle picture is showing you. If it's isotonic, then the plant cells will be flaccid and the plant will be kind of wilting a little bit. Better give it water quickly or it's going to die. If plant cells are surrounded by a hypertonic solution, this is really bad news for the plant. The plasma membrane will pull away from the cell wall. Take a look at the figure, it shows you this. It's called plasmolysis. And the plasma membrane is shriveling away from the cell wall. If that happens, the plant cell is going to be dead. It's like past the point of no return. So if you get to a plant that's just like totally wilted over, looks pathetically wilted, there's a good chance that those cells are now surrounded by hypertonic solutions, and they're not going to be able to recover no matter whether you water your plant or not. Then that's going to result in death for the plant. So this gives you a comparison of how cells with cell walls respond a little bit differently to hypo, iso, and hypertonic solutions compared to cells without cell walls, like animal cells or a paramecium.